Pregnancy, how a wonder is born. Pregnancy is an extraordinary journey, a miraculous process where life takes shape and grows within. From the union of two cells to the development of a fully formed human being, each stage is a testament to the intricate and awe-inspiring mechanisms of life. This journey, unique to every individual, unfolds over 9 months, encompassing moments of anticipation, transformation, and wonder. Understanding the biological marvel behind pregnancy not only deepens our appreciation for this gift of life but also highlights the intricate interplay of science and nature. Let us explore this incredible process step by step and uncover how a new life begins and thrives. Men, and most significantly, women, possess extraordinary biological systems uniquely designed for the creation of life. At the heart of this process lies the ovary, a fascinating organ that holds around 500,000 eggs, each nestled within a tiny structure called a follicle. With each menstrual cycle, multiple follicles are stimulated to mature, but only one typically dominates. This dominant follicle grows to approximately 2 centimeters and moves toward the fimbriae, the fringed end of the fallopian tube. During ovulation, the fallopian tube skillfully captures the released egg, marking the beginning of an incredible journey. The egg itself is a masterpiece of biological engineering. Its outermost protective layer, the corona radiata, shields it from the external environment, while an inner layer, known as the zona pellucida, serves as an additional barrier. For fertilization to occur, these layers must be successfully breached by a sperm cell, a task requiring precision and determination. Within the egg's nucleus lies a treasure trove of 23 chromosomes, each carrying the DNA that acts as a blueprint for building a human being. This genetic material determines characteristics ranging from physical traits like eye color and body structure to intricate cellular functions. When abnormalities occur, such as the presence of an extra chromosome, conditions like trisomy can arise, underscoring the precision required during this process. Meanwhile, the egg begins its journey through the fallopian tube, propelled by the rhythmic motion of tiny hair-like structures called cilia and aided by mucus secreted from glands within the tube. Additionally, muscular contractions of the tube walls help propel the egg forward at an astonishingly steady rate of 3 to 4 millimeters per minute. While the egg advances, sperm cells, thousands of microscopic swimmers, are making their way from the opposite end of the fallopian tube. Each sperm cell's sole mission is to reach the egg. Guided by chemical signals released by the egg, the sperm cells navigate through the female reproductive tract in what is nothing short of a biological race. The ultimate goal for each sperm is to penetrate the egg's protective layers and merge with its nucleus, thus combining genetic material to form a new life. When a sperm cell successfully penetrates the corona radiata and zona pellucida, a cascade of events is triggered. The sperm's acrosome cap, a specialized structure containing enzymes, breaks down, allowing the sperm to enter the egg's cytoplasm. This marks the moment of fertilization, which activates the egg. Activation initiates structural changes in the zona pellucida, ensuring that no additional sperm can enter, thereby protecting the developing embryo. This process also prompts the egg to complete its second maturation division, producing a female pronucleus and a polar body. Simultaneously, the sperm's tail and head disintegrate within the egg cytoplasm. The sperm nucleus then undergoes dramatic changes, enlarging and decondensing to form a male pronucleus. Within this nucleus are 23 unreplicated chromosomes containing the father's genetic contribution. As the male and female pronuclei draw closer together, each duplicates its DNA, creating replicated chromosomes in preparation for their union. When the pronuclei dissolve, the chromosomes from both parents align within the spindle apparatus in a process called syngamy. At this point, the single-celled zygote is formed, marking the origin of a new human being. The cleavage process. The journey of the zygote continues with a series of rapid cell divisions known as cleavage. The zygote divides into 2 cells, then 4, then 8, and so on, forming a compact cluster of cells. By the time it reaches the 16 to 32 cell stage, the structure is called a morula, named for its resemblance to a mulberry. These cells remain enclosed within the zona pellucida, and with each division, their size decreases while their number increases. Eventually, a hollow structure called the blastocyst emerges. The blastocyst consists of a fluid-filled cavity and 2 distinct cell groups. Embryoblast, this cluster of cells will develop into the embryo, the initial stage of the new human being. Trophoblast, these cells will give rise to the placenta and protective membranes, playing a crucial role in sustaining the pregnancy. The journey to the uterus. Fertilization typically occurs at the far end of the fallopian tube. The first cell division happens approximately 24 hours after fertilization, setting the stage for the morula's development. As the morula continues to grow and divide, it is transported toward the uterus. By the time it reaches the uterine cavity, it has developed into a blastocyst, ready for implantation. The uterine lining, or endometrium, undergoes remarkable preparation to welcome the blastocyst. Rich with blood vessels and glands, it provides a nourishing environment for the developing embryo. However, before the blastocyst can implant, it must hatch from the zona pellucida, shedding its protective shell to embed itself into the uterine lining, much like a tiny seed planting roots in fertile soil. Implantation. Implantation is a critical step in early pregnancy. The trophoblast cells at the outer edge of the blastocyst fuse with the endometrium, forming a specialized structure known as the syncytial trophoblast. This structure invades the uterine lining, facilitating the exchange of nutrients and waste between the mother and the developing embryo. Meanwhile, the embryoblast differentiates into two layers, the hypoblast and the epiblast, which will further develop into the embryo and its supporting structures. As the embryo burrows deeper into the endometrium, rapid cell growth occurs, often leaving a small wound that may result in harmless implantation bleeding. The process also establishes essential structures such as the amniotic cavity and yolk sac, which play vital roles in nourishing and protecting the embryo. Around the growing embryo, a large fluid-filled space called the chorionic cavity forms, surrounded by the expanding trophoblast. This initial stage of pregnancy sets the foundation for the extraordinary development to follow. From a single fertilized cell to a complex multicellular organism, the journey is nothing short of a biological marvel, demonstrating the intricate beauty of life's beginnings.